Something is changing in the sea, threatening some of our planet's most spectacular treasures.
They can move, they can adapt, or unfortunately, they die.
And attacking the ocean's fundamental web of life. Scientists are in a race against time to discover the real risks and come up with solutions. Off a remote island, an amazing discovery. A spot that could hold the key to our future.
We really hit a jackpot. This is such a scientific gold mine, like nowhere else in the world.
Can we crack the code of a rapidly changing ocean to understand what it means for life before it's too late? Lethal Seas, right now on Nova.
Major funding for NOVA is provided by the following. The David H. Koch Fund for Science, supporting NOVA and promoting public understanding of science. And the Corporation for Public Broadcasting. And by PBS viewers like you. Thank you. Additional funding from Roger and Vicki Satt. Additional funding for Lethal Seas is provided by the Candida Fund, furthering the values that contribute to a healthy planet.
In a remote corner of the sea, a natural wonder, a coral reef filled with exotic creatures. Reefs like these aren't just stunning to look at. They're some of the most fertile places on Earth. Home to about a quarter of all species living in the seas. Now, this abundance of life is under threat by an invisible killer. One that's taking its toll, not just here, but in our own backyard. Off America's northwest coast, the riches of the sea look like this. Pacific oysters. A keystone of a $250 million shellfish industry. But not long ago, a mysterious threat emerged from the depths, putting the oysters and all who depend on them in jeopardy. Including oyster growers like Mark Weigert. Mark and his wife Sue Cudd run an oyster hatchery. In giant tanks of seawater, they grow baby shellfish called larvae, the foundation of the region's oyster population. Business was booming until recently.
We first noticed a real solid change in 2007. We actually could not produce any larvae at all, and that definitely gets your attention.
All of the larvae in the whole place were pink. They were not feeding, they looked terrible. We just knew they were all gonna die.
For months, the couple struggled to solve the mystery of the dying baby oysters.
We saw behavior in the larvae that we'd never seen before. They would circle around and then just kind of lay on the bottom. We call them lazy larvae. They'd actually dissolve in the water, and we didn't know what was going on.
Sue hunted for clues under the microscope and found some disturbing evidence. This is a magnified image of a healthy larva, a one-day-old oyster. It's already started producing its shell. And this is what Sue was seeing in her microscope. The shell was deformed and pitted. The question was, why? The couple searched for a possible killer, but they never suspected that the culprit could be the seawater itself, specifically the water's pH.
I think the initial reaction from a lot of people was, don't worry about the pH of the water. That's never going to be a problem. It was a problem. The pH was dropping too low.
pH is the standard measurement for acidity. The lower the pH, the higher the acidity. One of the hatchery workers suggested they run a simple pH test on the water in the oyster tanks. Water pumped from the bay. The results were shocking. The acidity of the bay water was a staggering 6 times higher than normal seawater. The baby oysters couldn't handle it.
What we've realized about the chemistry of the water is that it's everything. If the chemistry is not right, the larvae can die, they can stop eating, they can stop growing. Basically, you're not able to grow larvae.
With the mystery solved, the hatchery has found a temporary solution to the problem. Soda ash, a common water softener, lowers the acidity back to a level most of the young oysters can tolerate. Now, enough baby oysters survive and grow so that Mark and his crew can transfer them out into the bay, but at a huge cost.
We pump 75 to 100,000 gallons a day, and that's a lot of making your own special seawater. And there's goodies in the water the larvae like that I don't even know about that, you know, Mother Nature provides. So how do you duplicate what they like? It's a very tough question.
Mark and Sue are not alone in their struggles. Fisheries throughout the Northwest are having the same problem. Water chemistry that's changing, shellfish that won't grow. And scientists are finding it's not just here. Samples taken around the globe over the last 40 years reveal that average ocean acidity is increasing by about 5% every decade. Why is this happening? It turns out the driving force of ocean acidity is the same thing that's responsible for global warming. When we burn fossil fuels, they release carbon dioxide gas, or CO2, into the atmosphere. Much of that CO2 blankets the planet and raises the global temperature. But about a quarter of that gas is absorbed into the oceans. At first, scientists thought this was beneficial, since the more CO2 that goes into the ocean means less in our atmosphere, reducing the warming effect.
Oceanographers knew for years that carbon dioxide from the atmosphere was absorbing into the oceans. And everyone thought, oh, perfect, this is a service that the ocean is performing for us, storing the gas that we're releasing as it's a pollutant, it's a product of our activities. And so everybody thought, ooh, this is awesome.
But scientists soon realized that this service comes at a cost. The extra carbon dioxide absorbed by the ocean is dramatically changing the water's chemistry.
We're dissolving 30 million metric tons of carbon dioxide into the surface of the ocean each and every day. And that is what's driving down the pH, or increasing the acidity.
This is how it works. When carbon dioxide dissolves into the ocean, it reacts with water, H2O, to form a weak acid called carbonic acid. This almost immediately breaks apart, releasing hydrogen ions. Those are hydrogen atoms stripped of their electrons. This is what increases the water's acidity, and it can have a devastating effect on marine life, like oysters. Healthy baby oysters are busy building their shells. They pull the ingredients they need from the surrounding water, including calcium and a substance called carbonate, combining them into a tough matrix, calcium carbonate. But when too much carbon dioxide gets into the mix, the resulting acid can radically alter one crucial ingredient, carbonate.
There's plenty of calcium in the ocean, the carbonate is kind of what we call the limiting ingredient. There's not a lot of carbonate.
When acidity rises, and there are lots more hydrogen ions floating around, they react with carbonate, taking away this essential molecule.
Instead of carbonate being all over the place, they're few and far between. And so what happens is, physiologically, it's just much more difficult to make shell.
Without enough carbonate in the water, the baby oysters simply can't build their shells, and as a result, they die. It's not just oysters that need carbonate to survive. The changing chemistry of the ocean threatens all kinds of shelled creatures. Scientists now face an urgent challenge: to understand what this will mean for life in the sea. Along with the oysters, how many other species will struggle to survive?
From a biological standpoint, predicting what will happen to individual animals, populations, and ecosystems is incredibly difficult.
I don't think right now we have a clue how that's going to change the development of any given species, and then, well, how one species grows up influences its prey, and it influences its predators, and it influences its competitors, and so it is really complicated.
The problem is most urgent here, on the world's coral reefs. These are some of the most spectacular places on Earth, filled with exotic creatures. Coral reefs occupy less than 1% of the ocean floor, yet they provide a home for at least a quarter of all marine species. And they're a key resource for survival for an estimated 500 million people. The foundations of these beautiful reefs are made of the skeletons of countless corals, built up over thousands of years. Like the oyster shells, these skeletons are made of calcium carbonate, sometimes known as limestone. And this makes them especially vulnerable to the ocean's rising acidity.
Corals are incredibly sensitive to high carbon dioxide conditions because they're forming a skeleton that is made out of limestone, and limestone and acid just don't go together.
Katerina Fabricius has spent a lifetime diving on some of the world's most pristine reefs.
It's just one of the most amazing ecosystems in the world, and seeing them degrading over time is just a real shame. It's also a moral and ethical responsibility for us to protect the reefs for our children and our grandchildren.
Many coral reefs worldwide are already suffering because of warming seas. How will the changing ocean chemistry affect them? Katerina believes she's discovered a clue. A very special reef located in remote Papua New Guinea.
Side is the straight stretch on the reef flat here. We'll be able to see the bubbles from the surface.
Set among the volcanic islands of Milne Bay lies one reef that Katerina found by accident more than a decade ago. It's a place where the water looks a bit like champagne, filled with streams of gas bubbles rising up through the sea floor, caused by volcanic activity. Samples reveal they are pure carbon dioxide. And just like the carbon dioxide that's mixing into the oceans from the atmosphere, these bubbles react with water to form an acid, lowering the water's pH.
It was just so exciting. I realized maybe we can use that as a natural laboratory.
There's so much carbon dioxide, the acidity is much higher than normal. In fact, the water chemistry here today is similar to what scientists think could happen to the rest of the ocean in 50 to 100 years if carbon dioxide levels continue to rise. So it could offer a unique window into the future of coral reefs across the globe.
So I thought, bingo, that's it. We gotta go back. We are going to the site Ili Ili Bur Bur, which is the best bubble site we've got.
Katerina and her team are now into their 4th year of study here. One of only a handful of coral reefs in the world with naturally high acidity.
My hope is very high that these guys here are cracking it and producing the world-class science that is so needed to understand ocean acidification.
For the team, these waters offer a unique opportunity to compare 2 marine environments very close together. The control site, where the ocean chemistry is normal, and the bubble site, where carbon dioxide is dramatically raising the water's acidity. How is life here, the corals, and the creatures that depend on them, responding to the changes? Are some adapting to new conditions? How many are struggling to survive? With just 2 weeks to get the work done, the team dives into their experiments.
Each of us is focusing on 2 or 3 different projects. I'm looking at how the conditions for corals is changing. That is stuff we cannot do in the laboratory.
Corals are unusual creatures. Each of these branching clumps is actually a colony, made up of thousands of tiny building blocks called coral polyps.
This is all one organism, but like a plant having individual leaves, the corals have individual polyps that are feeding separately from each other. They can share the food, but each polyp can respond to plankton touching its tentacles and grabbing it. Once they're digesting it, they're spreading the juices across the colony.
Corals are happiest and healthiest where the water is clear and well lit by the sun, and where they don't get too much competition from seaweed. But their unusual lifestyle makes them especially vulnerable to a changing environment.
Corals are actually animals, but they're glued to the ground like a plant is. They're not able to pack their bags and move to another side if the conditions is no good for them.
Where the carbon dioxide bubbles are most intense, few corals seem to be finding the conditions favorable. These large, extremely tolerant corals are called porites. They have thick skins and now dominate the reef. The colorful branching corals and soft corals that flourish in most coral reefs are gone. The team works around the clock in all kinds of weather. Some days, nothing goes according to plan.
I think there's some problem with one of the generators. One of the exhaust pipes had a bit of a breakdown.
Underwater conditions are difficult.
This site we have massive rocks, so it's really hard to drill inside the structure.
And some of the coral experiments have become dinner for some hungry snails.
At least 50% of little coral branches we made had been eaten by these snails.
Yeah, not happy. Experiments by nature quite often fail. It's requiring a lot of patience. But eventually we'll sort it out and we'll find some other ways to test the same questions.
While Katerina is studying the impact of high acidity on the corals themselves,
So I'd probably first jump in.
another team member, Laetitia Plaisance, is studying the many other sea creatures who depend on them. Where the water chemistry is normal, the coral gardens of Papua New Guinea are among the richest on the planet. Home to more than 500 species of coral. And hundreds of thousands of different kinds of tiny sea creatures. The entire ecosystem rests on a coral foundation built from calcium carbonate.
Everything is a balance in the ocean. The reef is made of calcifying organisms, so that means they need higher pH to be able to build their skeleton. And this skeleton then provides the structure of the reef where all the other associated species can find shelter, and they can mate, and they can feed, and they can hide in that structure.
Today, Leticia is focusing on one particular habitat, the dead coral head.
I'm on my way to find a coral head, a nice dead coral head.
Living coral heads provide homes for tiny fish and many other animals. Even on normal, healthy reefs, coral heads will eventually die, but their calcium carbonate structure remains, and just like a dead tree, new creatures move in. Leticia collects dead coral heads from both the normal site and the bubbly site, where acidity is much higher and the pH is lower. Will she find the same variety of species living in both locations? To find out, she cracks open the coral heads, hunting for the creatures hidden inside their nooks and crannies.
I'm searching for the crabs and the mollusk, and here is a little ophiorid trying to escape. Another sea cucumber, I think, here. All part of a food chain, and if you miss a link in the chain, the rest of the chain can continue, can't go on. So yes, all those little, pretty insignificant species are extremely important.
When she takes apart the coral head from the control site, with normal pH and acidity, she's impressed by the number of creatures she uncovers.
This morning we collected a nice dead coral head from the control site, which meant regular condition. It was a beautiful reef, and when I opened the coral head, it was filled with a life, abundance, and diversity. So we have plenty of different species of crustaceans here. Squat lobsters and crabs and shrimps.
But examining the coral head from the bubble site, even before Leticia breaks it open, she is concerned.
The first thing that I noticed with this coral head is that it's covered with this type of algae. And the problem really is that this type of algae does not hold much diversity, which I'm used to see in other sites where the pH is much higher.
The collection of life looks more limited, but to make sure, to get an exact inventory of all the different species, Leticia analyzes the DNA of the samples she collected. For example, two tiny shrimp may look alike, but they could actually be different species. A DNA test will reveal precisely how many different species live here. The DNA results confirm what Leticia suspected. The highest species diversity is at the control site, where the acidity and pH are normal. But as the acidity goes up and the pH goes down, the number of species drops dramatically. For each type of animal, only one or two species are tolerating the high acidity, and the rest have disappeared.
So you have the same kind of shrimp 100 times, when in the regular pH conditions you have 100 different species.
Leticia finds that the high CO2 site has 30% less diversity of small sea creatures, like hermit crabs, shrimp, and starfish, than the control site. For Leticia, this is troubling news.
The reason why this group is very important is because it's the basis of the food chain in coral reef. The fish feed on those tiny creatures. So if those tiny creatures disappear, the fish community is going to change and evolve as a result of this loss.
It appears that changes in acidity aren't just affecting the corals themselves, but hundreds of species living at the bottom of the food chain. And if those at the base of the food chain are at risk, so are all the creatures that rely on them for survival. These kinds of changes aren't just happening at coral reefs. cold water absorbs more carbon dioxide than warm, so acidity could rise in the Arctic and Antarctic seas more quickly. And that could mean trouble for the creatures here that, like coral and oysters, rely on carbonate to build their shells and skeletons. Aboard the Aurora Australis, the Australian Antarctic Division's research vessel, scientists are studying tiny plankton, some the size of a pinhead.
So we get to throw that back over the side.
Donna Roberts has spent much of her career in the Southern Ocean, and her favorite kind of plankton are tiny animals called pteropods. These almost transparent sea snails swarm in vast numbers in the colder waters of the Arctic and the Antarctic oceans. They are at the bottom of the food chain, providing food for whales, fish, and ultimately us.
The pteropod shells are gorgeous. It's just like a garden snail, but instead of having one slimy foot, it's evolved to live in the ocean with wings. Scientists call pteropods the potato chips of the sea because they're meaty and a lot of things eat them, but they've got that added little crunch.
Tucked away in this cold room at the University of Tasmania are pteropod samples from Antarctica, dating back to 1997. Donna is analyzing their shell weight to see if rising acidity has affected them.
It does matter if a little organism is losing weight because it's a measure of how healthy they are. So if their shells become thin, then they're going to put more of their energy towards building what little shell they can than reproducing.
Pteropod shells collected today are 35% smaller and more fragile than those caught a decade ago. Yet another sign of rising ocean acidity.
So this is a healthy set of shells that we collected in 2002, and I'll just show you for comparison what unhealthy shells look like. So you can see this one here has lost a segment of shell, and this one here is incredibly thin, very paper thin. Shells like this often fall apart in my hand when I'm trying to weigh them.
Scientists say that, like the baby oysters, there could soon come a point at which the levels of carbon dioxide will be too great for these beautiful little sea butterflies.
Most people have not heard of what a pteropod is, but they are vastly important. They're holding up the entire Southern Ocean food web in places.
Pteropods are a key food source for lots of animals that swim the seas. Not only in the southern oceans, but in the northern oceans as well. So what would happen if pteropod populations decline, or even disappear around the globe because of rising acidity?
The ecosystem effects of removing all the pteropods are just completely unclear. If we take them all away, we really don't know what the effects might be, but they may be quite dire for the things that feed on them.
Among the things that feed on pteropods are things we feed on, like salmon.
If you're a pink salmon, you can't just remove 10–15% of a juvenile pink salmon's diet and expect it to, just, oh, I'll just eat a little more of this instead. It's not evolutionarily hardwired to do that. So when you start removing huge components of what other individuals eat, what other species eat, that's a problem.
If higher acidity reduces or wipes out populations of creatures at the bottom of the food chain, the consequences could be bad for those further up. The worst possible scenario would involve a chain reaction of extinction, resulting in mass die-offs throughout the oceans. It has happened before. The fossils that trace the history of life in the ancient oceans reveal dramatic moments when suddenly many living things went extinct.
We think many of the big extinction events in the past have been related to some rapid event, whether it was an asteroid impact or some rapid, large increase in volcanism. Temperature was changing, ocean chemistry was changing. That's where we see the big extinction events.
The most famous extinction was 65 million years ago, when the dinosaurs disappeared. But an even bigger catastrophe hit the oceans 250 million years ago, when the Permian era ended. The leading theory is that massive volcanic eruptions caused carbon dioxide levels to spike quickly. The result was what's now known as the Great Dying, when up to 96% of all marine life went extinct, including many coral reefs. If acidity changes slowly, many creatures may be able to adapt and evolve to cope with the new conditions. But sudden changes are often dangerous for life.
On evolutionary timescales, organisms are adapting to their environment. The environment's changing and life is always changing. The more rapid that change, the harder it is for the organisms to adapt.
And today, the burning of fossil fuels is releasing so much carbon dioxide, it's changing the environment of the ocean very rapidly. Scientists fear that if carbon dioxide continues to rise at high speeds, we could have a major extinction event on our hands.
The rate of change is happening much, much faster than these plants and these animals can adapt. Unless it happens at a much, much slower rate, evolution will not have an opportunity to keep up with the change.
Perhaps nowhere on Earth are the plants and animals of the sea more at risk than on coral reefs. 500 million people rely on these unique ecosystems for their survival. The fish that live around reefs form a major part of the human diet. So, how will rising ocean acidity affect these animals? In addition to messing up the base of the food chain, will the changing chemistry have any direct effect on the fish's own bodies? Researchers like Danielle Dixon are trying to find out. Until now, most of her work has been in the lab. But today, she's joining the team in Papua New Guinea, where at one unique reef, carbon dioxide bubbles are raising the water's acidity to extreme levels.
The thing about this location that makes it really, really interesting is that these fish have been living in this CO2 situation for a very long time.
You can see where the CO2's coming up there. Yeah, so that's the CO2 seep site.
Philip Munday is Danielle's former professor and collaborator. In experiments back at the lab, they exposed fish to high levels of CO2 and saw alarming results. The damage wasn't done to the fish's skeletons, but to their brain chemistry and their behavior.
What we do see that's really dramatic is the changes in their behaviour. So things like how they respond to chemical cues in the water changes, their response to sound changes. And what's really cool here is that we can come to these seeps, we can see fish that have probably been living for months, maybe even years under a high CO2 environment and see if their behaviour is still affected.
The team wants to find out if the behavior they saw in the lab will be repeated in the wild, where some fish have grown up in high CO2 conditions throughout their lives. They'll compare the behavior of the fish living both near and far from the carbon dioxide bubbles. First, they'll test how they respond to chemical cues in the water. In other words, their sense of smell.
Baby fish in particular have very good noses. It's not on the end of their face, but they have nostrils and water comes in and there's sensory cells inside the nostril and then water goes back out. Very much the same as we do.
Just as land animals use their noses to detect certain molecules that float in the air, fish noses can sense molecules moving through the water. Their sense of smell is critical for their survival. They use it to catch food, find their way home, and even to sense danger.
There's a lot of things in the water that produce a lot of smell. There's predators in the water that are trying to feed. There's prey in the water that things are trying to feed on. The corals themselves, they're also releasing chemical cues. Pretty much everything uses chemistry underwater. So yeah, reefs would be a pretty smelly place.
The first step for this experiment is to catch some fish.
They're very easy to catch. Just a little bit of clove oil, and then you can use your hand to waft them out of the coral, grab them in a net.
Next, they must catch a predator fish.
So got a fish?
Danielle wants to test the smaller reef fish's ability to identify their predators, like this rock cod, by their smell. A crucial survival strategy for animals on land or sea.
This fish is going to be used to make predator odor. I'm just going to soak it in some water, and then the cues from the fish will come out in the water, and I'll be able to test little fish to see if they can identify the predator cues over other cues.
For this experiment, water with the predator odor is being pumped into the top side of the flume. The bottom side has normal offshore seawater.
The fish that we have in the flume right now is taken from the control reef where no CO2 bubbles are happening. The fish identifies the predator through smell alone and stays in the offshore water side where no predator smell is.
The next fish has grown up in the carbon dioxide bubble site, where the water acidity is higher.
Now the fish is sitting in the water that has the predator scent.
Instead of being repelled, the fish seems attracted to the dangerous smell.
We've found that the CO2 is affecting the fish's cognitive ability, so it can identify the smell of a predator, but something's wrong with its brain, then it's having a hard time identifying it as a bad thing. It's just identifying the smell. After testing dozens of fish,
Danielle finds that the fish that grow up in higher acidity don't avoid the predator smell. Behavior that makes it much more likely that they'd end up as somebody's dinner. In addition to smell, Danielle wants to test another survival instinct, the ability to hide. On these reefs, many fish live among the corals, using their branches for protection.
These fish are like candy on the reef. They really like to be eaten. It is in their best interest to be closer to the habitat so that they can retreat back to it and they're not picked off as much by predators.
Danielle tracks how adventurous the little fish are. Which ones will play it safe and stick close to the coral shelter? This one is from the control site, where the conditions are normal.
You can see it retreats right into the coral. It might be exploring the habitat of the coral, but it's not really exploring the tank. The fish will stay like this for the entire trial in general.
The next fish comes from the carbon dioxide bubble site.
You can see it's checking out the water a lot. It's going quite far from the coral. It's not really retreating back into the coral. Being bolder could make you grow faster and get bigger, but it also makes you a lot more susceptible to predators.
Danielle tests several fish. The small fish living in the higher acidity are consistently more adventurous, behavior that could put their lives at higher risk. Researchers don't know yet how all the different kinds of fish that live in the sea will be affected in higher acidity. Some are likely to cope better than others, and some may find some surprising benefits. At the bubble site, one form of life that's thriving is the seagrass.
Seagrasses can live in very high CO2 environments. They actually like it, because seagrasses, like the terrestrial plants, they do take in the carbon dioxide and convert it into sugars.
For plants, carbon dioxide is a fuel. They use sunlight to turn it into sugar, leaving oxygen as a waste product. The rich supply of carbon dioxide here is fertilizing these seagrass beds. The thick lawn of seagrass appears to be an important nursery for baby fish. Biologists are hoping that many of today's fish will find a way to survive in an ocean with more carbon dioxide and higher acidity. Their bodies may be able to adapt to changing conditions, possibly by turning on particular genes.
There are mechanisms that might allow them to adjust to a high CO2 world. Some individuals will have better genes for that particular environment, so they pass their genes on and you get genetic adaptation.
Marine biologists are eagerly hunting for any signs of genetic adaptation to a rapidly acidifying ocean. Today, the race is on to find creatures that carry genes that would help them survive. And the best places to look are the ones where ocean acidity goes up and down. On the West Coast, from Washington State down to California, there's a seasonal upwelling of carbon dioxide from the deep ocean, lowering the pH and raising the acidity. Gretchen Hoffman is trying to find out how the marine life here copes with a fluctuating pH.
One thing that's unique about the California coast here is that we have upwelling. And this is where wind blows across the surface of the oceans starting in the springtime. And this wind brings up deep, cold, CO2-rich water to the shores. And that water has a really low pH. And this low pH water has an impact on the biology on the shores.
One creature that seems to have adapted to the ocean's low pH and high acidity here is the spiky sea urchin. These are the lawnmowers of the sea. They play a vital role keeping ecosystems healthy by devouring seaweed, which, without the urchins, could grow out of control. Unlike the oyster, which is struggling to build its shell in the high acidity of the northwest coast, the sea urchin seems to be flourishing, even though it sports a shell constructed of the same material, calcium carbonate. Gretchen believes the key to its success may lie in its genes.
We found that populations, say from the north in Oregon, that are really tougher in the face of these pH changes, they were expressing genes in a different way than sort of the wimpy Californian urchins were, that they were turning on more genes that would help them make their skeleton.
By turning on certain genes, the northern sea urchins became more efficient at building their calcium carbonate shells, even when the surrounding water had higher acidity. Gretchen wonders if this life-saving gene could be passed on to the more vulnerable urchins in the south.
In 4 or 5 spots, that looks great.
To find out,
Really good.
she sets up a crossbreeding experiment. The sperm from both northern and southern males is used to fertilize eggs from the less hardy southern females. Once the larvae begin to mature, Gretchen compares the two samples.
Now we're interested in measuring their skeleton. And the way I think about a sea urchin larvae is that it's like a freestanding backpacking tent. So the skeleton are actually like the tent poles. That's the thing that's made of calcium carbonate, and that's part of their body that could be challenged to form under ocean acidification conditions.
Amazingly, the experiment reveals that the northern urchin's tolerance to high acidity is passed on genetically to the southern female's young larvae.
So what we've learned is that larvae that have fathers from the northern populations actually retain their body size. They stay bigger even in the face of low pH. So it does matter who your daddy is in this kind of experiment. We know now that there are populations that have genetics that will allow them to adapt, and we know how to look for them.
Researchers are looking all over for other marine life that is surviving in more corrosive waters, especially corals, since so far they seem to be some of the most vulnerable creatures in the sea. About 1,000 miles northwest of Papua New Guinea, off the Pacific islands of Palau, sits another coral reef where the acidity is naturally high. Here, there are no carbon dioxide bubbles rising up from below. The acidity seems to be caused by the density of aquatic life, combined with very slow water circulation in these shallow lagoons. Despite the high acidity, many corals here appear to be healthy. Ann Cohen is trying to figure out why.
We believe that we have found one coral reef community which, for some reason that we don't quite yet understand, it could be a genetic adaptation, or it could be a combination of environmental factors that allows them to survive and cope with this low pH conditions.
One important feature of this environment may be that in these shallow lagoons, the corals are extremely sheltered from any waves or storms. And that may be the key to their survival, so far. For marine scientists, places like these are precious.
We have to put those systems at high priority for protection. Those systems might be the only ones that are left at the end of the century.
Unlike this unusual spot in Palau, most reefs have to cope with powerful storms from time to time. And they rely on fast-growing corals to quickly rebuild their foundations. Back in Papua New Guinea, Katerina Fabricius would love to find evidence that the corals here, bathed in carbon dioxide bubbles produced by volcanic activity, are adapting, finding ways to be resilient in spite of the higher acidity.
Here, hopefully, some organisms have gotten used to the high carbon dioxide concentrations, which would be a good thing because it would tell us that maybe there is a future for coral reefs after all.
14 months ago, Katerina placed over 100 blank tiles at her study sites just in time for the annual coral spawning, hoping to compare the growth of baby corals in areas with normal and high acidity. The spawning of the coral is one of the most spectacular natural events on Earth. The corals synchronize their spawning with the lunar cycle, releasing billions of eggs and sperm, usually on the same night. Within a few days, the tiny coral larvae must find the right place to call home. Will these baby corals settle and grow on Katerina's tiles? Or will the high acidity ruin their chances? Now, she'll find out. The first tiles are from the control site.
They're looking fantastic. All the pink color is coral and algae, which are really important for the health of the reefs.
This pink coral and algae is often called the cement of the reef. It's one kind of algae the corals actually do like. Young corals are genetically programmed to zero in on it because it provides a strong foundation for their growth. But it's made of a type of calcium carbonate that will dissolve even faster than the corals if the pH drops too low. Katerina now moves to the high acidity bubble site and collects the tiles there. It takes hours to inventory exactly what's growing on all the tiles. The ones from the control site, where acidity is low, have cultivated a healthy batch of young corals.
The tiles from the control site already have got a fair number of large coral colonies established. Baby coral settled on the edge here is growing quite happily. So that's about 1 year old. Coralline algae here, the pink stuff. There's a speck here. There's one sitting here right at the edge. I found up to 19 coral settlers on this very small space. Not all of them will survive, but still it's very good for the coral reefs to establish.
But the tiles from the carbon dioxide bubble site tell a different story.
We found hardly any coral recruits on them. There's no pink left, no coralline algae. We're getting a lot of seaweed that are occupying the space for coral to settle. The fact that there's so few coral recruits is of real concern. I didn't expect the differences to be so stark.
The expedition's nearly over. The findings here have been discouraging. Along with the scientists, the locals are concerned about the future of their coral reef.
How does this carbon dioxide concentration affect the corals on the sea?
The coral reefs will still be there in 50 or 100 years' time, but they will look very different, is our prediction. There will be less of those branching corals, and there will be possibly less fish as well, which is of real concern because it's important for food.
Right now, carbon dioxide levels are rising rapidly. And if that keeps going, the acidity of the ocean will continue to rise as well.
The problem now is that you can't fence off a coral reef island or a fringing reef or a barrier reef and protect it from acidification. This is a global threat.
With coral reefs in many places already vanishing at a rate of 1 to 2% every year, finding a solution is critical. So far, evidence that coral can adapt is slim. And many marine biologists are asking, is it realistic to expect the creatures of the sea to adapt to an ocean that is changing so rapidly? Or should we go to the source of the problem? The rising carbon dioxide in our atmosphere and in the seas, caused by the burning of fossil fuels.
The best solution is to reduce the emissions to the atmosphere or capture the carbon dioxide before it gets into the atmosphere.
There's no way to save coral reefs without stopping greenhouse gas emissions. I mean, there is absolutely no practical solution to protect coral reefs from warming and from ocean acidification if we don't stop carbon dioxide emissions in the world.
If we know what's causing it, why don't we just fix the cause and not twiddle our thumbs for 20 or 30 years trying to sort things out?
And if carbon emissions aren't cut, many are troubled by the question, what will happen to life in the sea?
It's a big question mark, really, what the ocean's going to look like, but it is going to look a lot different, and it is not going to be as resilient as it is today.
We're sort of crossing our fingers that there's all these complicated web of changes and that somehow we're going to be happy with the outcome. And I just think that's a bigger risk than we ought to take.